Today, we are on site at the San Diego Convention Center in California at the 2018 ASH annual meeting. We have recapped the top news each day during the meeting, and soon we'll speak with Dr. Owen O'Connor on some encouraging lymphoma abstracts and Dr. William Wierda on exciting chronic lymphocytic leukemia studies. Welcome to OncLive News Network. I'm Gina Columbus. In acute myeloid leukemia, results of the Phase 3 QuANTUM-R trial confirm that single-agent quizartinib improves survival over salvage chemotherapy in patients with relapsed/refractory FLT3 ITD-positive disease. Quizartinib also demonstrated a favorable safety profile in this randomized controlled trial. Data from Part 2 of the STORM trial evaluating selinexor in patients with penta-refractory multiple myeloma show that the agent, when added to low-dose dexamethasone, has clinical activity with rapid and deep responses. The adverse events associated with this regimen can be effectively managed with dose modifications and supportive care. Selinexor could be a potential option for patients with penta-refractory myeloma who have already received available therapies. In chronic lymphocytic leukemia, Phase 3 data from the MURANO trial show that the combination of venetoclax and rituximab have a higher peripheral blood undetectable minimal residual disease rate versus bendamustine and rituximab in patients with relapsed refractory disease. This was also observed in high-risk subgroups. These data are the first to demonstrate the value of peripheral blood MRD as a predictive marker of clinical outcome for the fixed-duration chemotherapy-free regimen. Findings from the Phase 3 ILLUMINATE trial demonstrated that the combination of ibrutinib and obinutuzumab had a significant improvement in progression-free survival versus chemoimmunotherapy treatment with chlorambucil and obinutuzumab in patients with chronic lymphocytic leukemia. The benefit was also observed in a high-risk patient population. Additionally, response rates and depth of remission were also higher with the ibrutinib regimen, which was also found to have a tolerable safety profile. Here now to discuss some of the studies being presented in mantle cell lymphoma and T-cell lymphoma is Dr. Owen A. O'Connor, who is the American Cancer Society Research Professor, Director of the Center for Lymph Node Malignancies, and Co-Program Director of the Lymph Node Development and Malignancy Program in the Herbert Irving Comprehensive Cancer Center at Columbia University Medical Center. Hi, Dr. O'Connor. We're so happy to have you today.
Thank you. Thank you, Gina.
Sure. So let's first talk about what's going on in mantle cell lymphoma at this year's meeting. What are some of the things that you would like to mention?
I think there's little question that the BTK inhibitors continue to evolve and are a class of drugs that Michael Wang first reported on in the New England Journal of Medicine that demonstrated pretty remarkable activity. In Michael's earlier work, he showed an overall response rate of about 65% with a CR rate of about 20% with ibrutinib. As everybody on the planet knows by now, ibrutinib is a irreversible inhibitor of BTK and has demonstrated also very meaningful durations of benefit. On the heels of ibrutinib, a second generation BTK inhibitor, acalabrutinib, came along. Acalabrutinib is best distinguished from ibrutinib because it is felt to be a little bit more selective on the kinome compared to ibrutinib. And most importantly, it doesn't hit TEC, one of these enzymes that people have attributed the risk of bleeding to. Acalabrutinib was also active, but I think at this meeting, one of the things that's new is some of the long-term follow-up data for ibrutinib and acalabrutinib, which have continued to confirm very good durability of response and really no change in much of that toxicity profile that was originally reported. But there's a third generation BTK inhibitor, another covalently bound BTK inhibitor called zanubrutinib being developed by BeiGene. And so there is some interesting data with that drug. And that drug again is purportedly to be more selective against the relevant kinases of the kinome, not hitting TEC and some of those others that people attribute the toxicity to. But in some data presented at this meeting, it demonstrated a pretty high overall response rate of about 90% or so, 80-90%, with a CR rate of about 20%. And while that in and of itself seems interesting, the side effect profile for all the world looks like ibrutinib and acalabrutinib. There was really interesting data presented by Dr. Song from Peking University. And they did a study in China with about 84 patients, so a much larger single-arm kind of study. And they reported in a more heavily treated population than the first experience in the US, they reported an overall response rate of about 84, 85%. But what sort of stunned people was the complete response rate. And the complete response rate in that Chinese population was about 60%.
Wow.
So if you go back and you look at Michael's data from acalabrutinib and ibrutinib, those floated around 20%. And if you look at the Chinese data, it's nearly tripling that CR rate. And as you might imagine, the expectation is that by having more CRs or deeper complete responses, you're going to get much better progression-free survival curves and much better durations of response. So the big question is why? Why is it that the Chinese experience has a much better complete response rate? And there's lots of reasons. You know, people have talked about likely issues revolving around cytochrome P450 and metabolism and some of these important ethnopharmacologic determinants, but I think it remains to be seen. We didn't see a lot in terms of PK. We didn't see a lot in terms of kinome scans and other things. It remains an interesting observations. These are small studies, so it could be a fluke, but it's definitely an interesting scientific piece of information to follow up on.
Yeah, it is interesting, definitely. Yeah. Now, honing in a little bit more on acalabrutinib, we were seeing some more data with it as a single agent as well as in combination. Do you want to go into those data a little bit more?
Yep. So there was interesting data presented looking at acalabrutinib plus BR. And as most people are aware, there's been a massive shift, thanks in part to Matthias Rummel's data, where he demonstrated that BR seemed to be safer and as good as R-CHOP-based chemotherapy regimens in mantle cell lymphoma. So easier regimen. I don't know if it's a standard of care, but it's probably pretty close. So given the activity of the BTK inhibitors, it makes complete sense to try and deepen those responses. Now, some might look at this and say, you're just making soup adding all these drugs to a standard like BR. But in fact, there's probably a good rationale because in mantle cell lymphoma, achieving PCR negativity is probably important. And there's really not a lot of data in the context of these kinds of regimens and their ability to do that. We know from this meeting this year and last year that the ability to achieve PCR negativity in CLL is probably very important in terms of its association with long-term survival. And we know from the Nordic Lymphoma Study Group that those patients who are PCR negative for the 11-14 translocation do better. And if you have a relapse of the PCR evident disease, you can treat with Rituxan and they still do well. So we didn't see data on PCR monitoring in this study, but they looked at a hodgepodge of patients. Patients that had relapsed refractory disease, as well as patients that were treatment naive. And as you might expect, the response rates in the treatment naive patients were really good. Overall response rates in the 90% range and CR rates in the 70% range. And in the relapsed refractory population, still very good. Overall response rates of about 84%, CRs probably in the rate of 50%. So that represents a pretty promising regimen, and I think it does raise the possibility that it could be explored in a randomized phase 3 clinical trial of BR against acalabrutinib BR. But I would like to see some PCR testing and see if this is an important issue and see if in fact the addition of a BTK helps us by achieving greater levels of not just complete remission, but PCR negative complete remissions.
Yeah, very good points. Now, we're also seeing some long-term data with a BCL2 inhibitor, venetoclax. So what are we seeing with this agent now, and what does that tell us about the role that venetoclax has in the MCL paradigm?
Yeah, you know, Matt Davids has presented a lot of this data, and you know, as one of the guys that helped develop bortezomib 100 years ago, and you know, that was the drug, it's really interesting to see now all these drugs. And you know, Constantine Tam has begun looking at BTK inhibitors plus venetoclax, and you might imagine that just like in CLL, where we're really not so much talking about FCR or bendamustine in CLL anymore, people are pretty optimistic that we're going to achieve pretty durable complete remissions in patients getting combinations of venetoclax with BTK plus CD20. The same paradigm is evolving in mantle cell. Now, mantle cell is a little bit more complicated because it is a broad spectrum of disease. most of these studies have been happening in patients with Ki67 indices that are low. And so in mantle cell lymphoma, the Ki67 index, that measure of how proliferative the disease is probably the most important prognostic feature of the disease. It might correlate with the 17p11q in CLL. And so there's not a great description of what that patient population looks like in these studies, and I would like to see more of that as we go forward. But most of the studies are done in patients with more indolent mantle cell lymphoma, and so that's the case with venetoclax. And so what we know is that these combinations work well together. We're not really addressing how well they're working in the different risk populations, i.e. those that might have more advanced high proliferative disease versus those with lower proliferative disease. But venetoclax is yet another drug that's producing extremely high response rates. Back in the day with bortezomib, when we got 30% and 6 months everybody was ecstatic. Now that's old news and probably wouldn't pass muster today. Many of these drugs are hitting overall response rates of 70 to 80% with CR rates in the 54% range and progression-free survivals and durations of response measured in years. So the paradigm is changing, and as you can see, every year we talk about this, it's changing fast.
A lot of potential going on.
Right.
Yes. So let's move on now to T-cell lymphoma.
Yeah.
What is going on in the frontline setting?
Yeah, finally that disease that we as a medical community had neglected for quite a long time, or been addicted to using the treatments in B-cell lymphoma as the standards. Things are finally changing. And at this meeting, there were two interesting frontline studies. One was the Echelon 2 study. And the Echelon 2 study is a very interesting trial. It looked at adding brentuximab vedotin, which is a CD30 targeted antibody drug conjugate, to the CHOP backbone. So it was a randomized study, largest to date, looking at CHOP against cyclophosphamide, doxorubicin, brentuximab, and prednisone. So the trial, interestingly, required that the eligible population, be made up of 75% patients with anaplastic large cell lymphoma. Now that, of course, is the disease where brentuximab got its early approval, and it's a disease characterized by 100% expression of CD30. The rest of the other PTCL subtypes don't express CD30 to the same robust extent. And so the response rates for BV and ALCL led to its very rapid approval. So in this trial, 75% of the patients had ALCL, and only 25% had non-ALCL. So the naive observer might say, oh, you stacked the cards in your favor here by putting in the subtype with the greatest vulnerability to that drug. But in fact, that 75% was done for a regulatory purpose. It was in response to the EMA, who required a post-marketing randomized study with the drug to demonstrate to confirm its activity and regulatory approval. So while there's no denying that 3/4 of the population had ALCL and they're more sensitive, the study was positive for both progression-free survival and overall survival. So the first time we've ever seen that. Now the skeptics are going to say, what about the other subtypes? How much CD30 expression do I need to treat those patients? And it's interesting that the trial required patients to have 10% CD30 expression to get in. But when the FDA approved the regimen, approved only 11 days after the submission, so a world record.
Wow.
The FDA approved no level of CD30, just CD30 positive. And so it raises interesting questions. Is CD30 important in the predicting the response or associating response? But I think the data are the data. If you have a CD30 positive T-cell lymphoma, this ACE, Cytoxan, doxorubicin, BV, prednisone regimen is the new standard of care. Another study, not positive, is ACT-1. And ACT-1 is presented by Francesco D'Amore from the Nordic Lymphoma Study Group, and looked at adding Campath targeting CD52 to CHOP-based chemo. So they did a very good study looking at CHOP against Campath CHOP consolidated by stem cell transplant. And so that study in the beginning was wrought with all sorts of challenges, including CMV, viremia, and infections like fungal infections due to the anti-CD52. They made the appropriate dose adjustments and a lot of that toxicity went away, but the final results presented at this meeting show no difference in progression-free survival or overall survival for those two arms. So good news, bad news. At the end of the day, we've made an incremental step forward with Echelon 2.
Yes, absolutely. And now, overall, what are some other abstracts in non-Hodgkin lymphoma being presented at this meeting that you're excited about?
Well, staying on the theme of T-cell lymphoma, I think that there's a lot of exciting stuff happening. And Lorenzo Fauci from my team is going to share some data looking at epigenetic combinations in the disease, where we're now using these as frontline therapy. And we're seeing response rates in the 80 to 90% range, and some subtypes where 8 out of 9 patients are responding. So looking at combinations like azacitidine and romidepsin and some of these novel drug combinations in T-cell lymphoma are beginning to get some land legs, and they're beginning to evolve in a pretty favorable way. We heard earlier yesterday about the Augment study, and the Augment study was also a study I was involved in on the DSM committee for many years, and it looked to randomize in patients that had one line of prior therapy with follicular or marginal zone lymphoma, Rituxan versus Rituxan plus lenalidomide. And the study was presented yesterday and showed a substantial improvement in the progression-free survival. So that was also a very positive study suggesting that adding lenalidomide to rituximab really produced, and it wasn't a trivial small difference. It was a really, what I feel and most others believe, is a clinically meaningful improvement in the progression-free survival for patients with follicular and marginal zone lymphoma.
Oh. Well, Dr. O'Connor, this was wonderful. Thank you so much for sitting down with us and highlighting all of these great abstracts.
You're
Appreciate
welcome.
it.
Thank you.
Great, thanks. Next, we visit with Dr. William G. Wierda, who is head of the CLL section and executive medical director of leukemia at the University of Texas MD Anderson Cancer Center. Hi, Dr. Wierda. We're so happy to have you here today.
Good afternoon. Happy to be here.
Yes, good afternoon. So, we have heard about a number of trials being presented in CLL at this year's ASH meeting. So let's first talk a little bit about ibrutinib versus FCR in untreated younger patients. Why is this study important?
So there are 3 large randomized trials that are very important that are being presented at ASH this year. The first of which is the trial that you're referring to, which was a trial that evaluated ibrutinib plus rituximab versus FCR for previously untreated patients who were fit enough to receive FCR-based therapy. Half the patients were assigned to ibrutinib plus rituximab, half to FCR, and that trial showed an improvement in progression-free survival, favoring those patients who received ibrutinib plus rituximab, and also in overall survival, favoring those patients who received ibrutinib-based therapy. So that is a notable feature because that's, of the 3 large randomized trials, that is the only trial that's showing ibrutinib being associated with improvement in overall survival. We were expecting it to be a positive trial with regard to progression-free survival. We need to hear the data and the details about the deaths that occurred on the FCR arm to have a better idea about why they are seeing an improvement in overall survival. However, it's a very significant and notable trial. The other notable feature about that trial is there was an improvement in progression-free survival, particularly in patients who have an unmutated immunoglobulin gene. So that progression-free survival difference was statistically significant. In contrast to patients who have a mutated immunoglobulin gene, there was not a significant difference in outcomes for those patients. So we've reported data, and others, other large trials have reported data in terms of a plateau for patients who have a mutated V gene, potential cure fraction with that population. So for me, we're still considering FCR an important treatment for patients who have a mutated V gene. For the patients who have an unmutated V gene, based on this trial, my impression is that, and my opinion is that ibrutinib is the standard of care. There are 2 other large randomized trials. One is a trial that's done by the Alliance that was presented as a plenary presentation. That was a 3-arm trial. patients were assigned to treatment with ibrutinib monotherapy, or ibrutinib plus rituximab, or bendamustine plus rituximab, another chemoimmunotherapy regimen. Bendamustine rituximab is important because it's tolerated by younger patients relatively well, but also older patients. So it's a little bit more intensive chemoimmunotherapy regimen than chlorambucil-based treatment, less intensive than FCR. And that 3-arm randomized trial showed improvement in progression-free survival for both of the ibrutinib-based arm, monotherapy and with rituximab. There was no difference in terms of overall survival, and there was no difference in terms of progression-free survival between the two ibrutinib arms. So there's still discussion, and the data does not necessarily indicate improvement in outcomes with the addition of a CD20 antibody to ibrutinib-based therapy if you're giving continuous indefinite treatment with ibrutinib.
All right, those are some great points there. Now, let's look at the prospective data from the Morano study looking at long-term outcomes of venetoclax plus rituximab in relapsed refractory disease. So what are the findings here?
So last year we heard the data from the Morano trial. This is a randomized trial of bendamustine rituximab versus venetoclax rituximab for previously treated patients. Last year the presentation reported improvement in progression-free survival and overall survival favoring venetoclax plus rituximab over bendamustine plus rituximab. The treatment with bendamustine rituximab is 6 cycles of chemotherapy and then patients stop. Venetoclax rituximab is a little bit longer treatment period, so patients can get up to 2 years of treatment with venetoclax, 6 cycles of the rituximab at the beginning of that treatment. But the question was whether, although there was a difference in progression-free survival, were those responses durable, because most of the patients that were reported last year were still on treatment with venetoclax. This year we received follow-up for 130 patients who had discontinued the venetoclax. And that data shows a durable response, and we're not seeing patients rapidly progressing after they discontinue venetoclax. So this follow-up is very important with regard to that, and there was also data presented in an oral session by Arnon Cotter, who gave updates with regard to MRD and showing the fact that patients who achieve an undetectable MRD status after they stop treatment with venetoclax have a longer progression-free survival.
And you yourself you're leading an abstract on MRD status with venetoclax and CLL. So can you walk us through that study a little bit?
Right, so that was an analysis of a combination of trials with venetoclax for relapsed and refractory patients. There were 2 trials that we included in that group. One was for relapsed patients with 17p deletion, and the other was for patients who were refractory to ibrutinib or idelalisib. And that was a continuous treatment with venetoclax monotherapy. So patients went on venetoclax and they continued until progression, and then they were discontinued at that point. That analysis shows that even with continuous monotherapy, depth of remission is important in terms of outcome. So with our landmark analysis, we demonstrated that patients who achieved an MRD undetectable status in the blood had a longer progression-free survival with a landmark at 12 months as well as a landmark analysis at 24 months. So clearly this shows improvement in outcomes for patients who achieve a deep remission, even in patients on venetoclax who are on monotherapy and continuous therapy.
Got it. It's very interesting. So now we're also learning about acquired resistance to venetoclax with the recurrent GLY101VAL mutation. So though this is also a late breaker, what are you looking forward to learning about this study?
So this is a late breaker. We haven't seen the data yet. There are patients who develop resistance to venetoclax, and we haven't had a great definitive answer in terms of what the mechanism for that is. People have looked at various things: upregulation of MCL1, upregulation of BCL-XL. Well, this late-breaking abstract has looked at mutations in BCL2 and has correlated refractory disease to venetoclax with development of this particular mutation in a subgroup of patients who they evaluated.
Great. Now, there's also been a lot of CAR T cell therapy abstracts going on in CLL. So what does this really say about what potential role this type of therapy has? What kind of data have we seen that's exciting?
Right, for CLL, the preliminary data that we were seeing with CAR T-cell therapy was somewhat limited, and the responses were limited. So the complete remission rate with CAR T-cell therapy in CLL had been reported at about 20 to 30%. And so the question is, how can we bring those response rates up, the complete remission rate up, to where we are seeing with ALL and lymphomas? And so the abstracts that were presented at ASH this year were strategies to enhance the complete remission rate. Things like adding ibrutinib to the CAR T-cell therapy. And that appears to be increasing the response rate.
Definitely. So now looking overall at some of the other CLL abstracts being presented this year's ASH meeting, what are some others off the top of your head that you're really excited about this year?
In terms of other strategies for CLL, I mean, the big news has been these large randomized trials. There are some other abstracts of newer agents that are being evaluated, but those are very preliminary data. So I think really the big news from this ASH is these large randomized trials and changing and shifting the standard of care for patients. We've presented data from MD Anderson looking at combinations of small molecule inhibitors. This was an update from an abstract that we presented last year. And that strategy, we're getting very deep remissions, and we're very excited about that strategy. And I think it gives a promise of fixed duration treatment. So it's treatment for a defined period, a year perhaps, and then discontinuation of treatment and patients remaining in remission for long periods of time. The question is, when they relapse, how do we manage them when they relapse? But clearly we're getting very deep remissions with these small molecule inhibitor combinations.
Great. Well, Dr. Wierda, thank you so much for sitting down with us today and walking us through all these CLL abstracts, and have a great rest of your meeting.
Thank you.
Thanks. That's all for today. Please visit OncLive.com to watch our prior episodes from this meeting. Thank you for watching OncLive News Network on location from the 2018 ASH annual meeting in San Diego, California. I'm Gina Columbus.